If you couldn't tell from the music, this is Group Tango, and today we will be talking to you about randomized controlled trials. We will start with a definition. A randomized controlled trial is a study in which subjects are placed randomly into one of several predetermined clinical groups. One group will be a control group that will be used as a comparison, while the other group, the treatment group, will be exposed to an independent variable. The control group. The control group will be subjected to a standard practice, no intervention, or may take a placebo. The control group should not know that they are in the control group in most cases, just as the treatment group will not be sure that they are receiving treatment. In some trials, this is not possible. For the treatment group will be exposed to an intervention or a treatment. The treatment group results will then be compared to the control group. The results can then be deemed statistically significant if the p-value is less than 0.05, if they reject the null hypothesis, or not statistically significant if the p-value is greater than 0.05.
Okay, so now let's talk benefits. So the advantages of RCTs are that they are a well-known type of trial, often referred to as the gold standard for clinically testing treatments and drugs. There's also the random assortment, which means there's theoretical elimination of bias, specifically selection bias and confounding factors. All intervention groups are treated identically, except for the experimental treatment, thereby reducing the likelihood that the risks or benefits are due to causes outside of the experiment. Finally, the results can be analyzed with various statistical tools.
The downfalls and limitations of an RTC begin with standardizing mock-ups. Treatments that are too invasive or surgically involved may be too difficult to realistically mock up in a placebo group. There's also difficulty in finding correct sample sizes. If there's too small of a population size, or the patients with rare diseases are too hard to find properly and study in that affected population, this will be hard to do. Recruitment sample sizes or specific populations that may be hard to find based on availability or other external elements such as money, time, travel, etc. will be hard to use with an RTC. Ethically, it may be not fair to give treatment to one population and not the other for a specific disease, especially when it is life-threatening or a critical illness is involved.
Now, an example of an RCT. The test objective. A cosmetic company is testing the effect of a new lotion containing vitamin E on the improvement of skin conditions. Study design. 100 trial participants were randomly separated into equal groups of 50, an experimental group and a control group. The skin condition, moisture, wrinkle, dryness for all participants were tested prior to the trial. Skin was categorized based on objective: poor, fair, good, and excellent skin quality. 50 participants were given a body lotion containing vitamin E to be used for 1 year. 50 participants in the control group were given a placebo treatment, body lotion with no active ingredients, to be used for 1 year. The results. The skin of all participants were re-examined after 1 year. The skin condition improved in 80% of the participants using the lotion containing vitamin E, and 10% of the participants in the control group. The p-value was 0.04 using a McNamar test used for nominal studies. The study found that there was a statistical significance between participants that used vitamin E lotion and those that did not use vitamin E lotion.
Thank you very much for watching the video today. We hope you learned a lot.
Cheers!